Okay, so this morning I'm going to go over some features of this worksheet, which I know is going to be confusing to everybody. So after we do that, we'll then go on and start talking about RNA therapeutics. So this is about basic practice in looking at these Western blots and things, and also the qPCR data to try to look at each piece and understand what's going on. So I'm going to take it slowly, one piece at a time. Okay, so we have an experiment here where we're looking at the effects of silencing or attempted silencing of ADAR1 and or ADAR2 in cell lines. Okay, so we're looking at Western blots on the figures A and B. So we're going to look at those first. So A, we have Western blots, and we have cell lines to which we've added either a silencing control, which is basically a random sequence, or a scrambled control, which is not expected to have any effect on ADAR1 or ADAR2. So that's our comparison. Then we're going to look at an attempt to silence or shut down the expression of ADAR1 using our Western blot at the bottom of the figure. And so we can see that in the presence of the silencer, intended silencer, ADAR1 levels are greatly reduced as compared to the control, which is why we have 3 significant stars above those differences. And the control here is normalized to 1, so our y-axis we can read is 0.2, so there's been approximately a 5-fold reduction in ADAR1. So that was the intended purpose of the experiment, to shut down ADAR1. Now, to make sure it's specific, that it's just shutting down ADAR1, not ADAR2, we're going to look at the effects of the silencing of ADAR2 on ADAR1. Now, we would hope that it would not have any effect because those two proteins are different. But this Western blot shows us, in fact, that it's true, that silencing ADAR2 had no effect on ADAR1, as you can see from the bar graph. So it's a matter of taking a look at the Western, taking a look at the bar graph, and try to interpret that in terms of what's going on. So this doesn't prove anything. It just tells us that when we try to silence ADAR1, we did in fact silence ADAR1 and not ADAR2. Panel B is basically the same idea, but in reverse. We're looking at the effects on ADAR2 of either the control, the silencer of ADAR1, or the silencer of ADAR2. So this also makes sense. We can see the Western on the bottom. We see 2 bands for ADAR2, which are not quite explained. And we have our control, where we're comparing it to, in this case, GAPDH, which is glyceraldehyde 3-phosphate dehydrogenase. And so that is set to 1. And we can see that silencing of ADAR1 in the middle lane has no effect on ADAR2, which is what we want. And then finally, we look at the effects on ADAR2 of silencing ADAR2, and in fact, it is silenced by about 5-fold. So that tells us that what we're trying to do is working, in that we try to silence ADAR1, we do it, it works, and it doesn't cross-silence ADAR2, and vice versa. Okay, so this panel basically summarizes what I just said, that our experiment is working as hoped. Okay, so panel B is the same, but in reverse. Again, what we're intending to silence is being silenced and nothing else, which is what we wanted. Okay, so now that we know that our silencing RNAs are working as hoped, we want to know if there's any effect on a different gene, which is to say the aryl hydrocarbon receptor, AHR. So in this experiment, we're not looking at protein levels. We're looking at RNA levels, comparing cells containing the silencing of ADAR1, ADAR2, and a control, their effects on the aryl hydrocarbon receptor RNA. So this is, again, normalized to the GAPDH, and the ratio there is set to 1. And then we look at, by comparison, there's no change in the aryl hydrocarbon receptor in the presence of either silencer, because our qPCR data are telling us that there's no change in the amount of RNA. Those bars look exactly the same. So there's no effect of either of those silencers on that receptor mRNA. Okay, here's where it gets interesting or a little bit confusing. So if we already learned that the AHR RNA hasn't changed, but now we're going to look at the effects of the protein levels. So again, we're going to start with our silencer control, where this time they're going to show us the amount of protein for both the aryl hydrocarbon receptor and the protein level. So we can going to set that ratio to 1 as a normalizer. And the slightly surprising result might be that in the presence of when you silence ADAR1, the aryl hydrocarbon receptor protein actually goes up, even though we know that the amount of RNA was exactly the same. In a similar experiment, we silenced ADAR2, and that has no effect on the protein. Right? So silencing ADAR1 somehow leads to an increase in the amount of protein produced from the aryl hydrocarbon RNA. So at first glance, that's a little hard to understand. Okay, so if you think about it, in this situation, the silencer control would represent the edited RNA, if in fact this RNA is edited by ADAR1 or ADAR2. So that's the key thing to notice here, that the aryl hydrocarbon protein is actually increased in the ADAR1 silencing, which means that the unedited ones are higher. So we can see that there's an effect on protein, not RNA, and the effect has to be on either translation or stability of that particular protein. Okay, so to explain this, we have to look at some effect on translation or of the protein itself, not the RNA, which doesn't change. So remembering that the unedited form is the one that is there in the silencing ADAR1 experiment, and that the control silencer would be the edited version. So we can conclude that the edited RNA either is translated less efficiently or is less stable than the unedited one. Now, either explanation is plausible, but given the effects of editing on translation, we're more likely to believe, most likely, that the effect is on actual translation of that particular RNA. Okay, so our real business of today is to talk about oligonucleotide therapies and how this can work for us clinically. So the oligonucleotides that are going to be used therapeutically to knock down RNAs, either at their level of their degradation or their function, we're going to be using different types of RNAs in different kinds of experiments. We might use double-stranded RNAs, modified RNAs, or even DNA oligonucleotides in some cases. So first, you always have to make sure something works in cell lines before delivering it to people. Once you get it to work in cell lines, you've got to work in animals, where its important aspect is how to actually deliver the RNA or the oligonucleotide to the right tissue at the right time. Delivery in people, of course, is a real problem, but things are improving radically. A naked silencing RNA of any sort doesn't last long in the bloodstream. It's degraded by nucleases filtered to the kidneys and may or may not get to whatever the tissue is where it is needed. So some experiments use most experiments really use modifications in the RNA backbone to try to make them less sensitive to nucleases. So the 2'-O-methyl does stabilize them, makes them insensitive to degradation, at least by certain nucleases. And the other substitution is a sulfur group in place of one of the oxygens on the phosphate in the phosphodiester bond. So those are both stabilizing modifications which allow the oligonucleotide to survive longer in the body, basically. So we're going to do a couple of examples. One is a oligo called Paterasan, which is for the treatment of TTR amyloidosis. This was a silencing RNA, and it was the first one to get approval from the FDA. It's a good one. Okay. Next time we'll talk about treatments for spinal muscular atrophy, a modified single-strand oligo. Okay, so Onpattro is a double-stranded, small interfering RNA, and it is delivered as a form of a lipid complex. As we've seen before, lipid nanoparticles are working out well as delivery agents in the human body. So the target of this particular oligo is a conserved sequence in the 3' untranslated region of a RNA encoding TTR, well, the transthyretin mRNA that is responsible for this particular disease. And you can see there's some, even though it's going to be encapsulated in lipid nanoparticles, there's still some of the characteristic modifications including 2'-O-methyls at the on the sugars. So this is a nucleic acid that has modifications in it, and notice the methylated sugars, but it's still put inside a lipid nanoparticle. So we've seen some of these already. The nucleic acid is incorporated into these special lipid particles. Now, other times disabled viruses are used, but we're not going to talk about that today. Sometimes there's actually the ability to directly inject into the site in the body where it's needed. Okay, so we've seen these before, lipid nanoparticles. The nucleic acids are encapsulated in these particles, which allow them to recognize sometimes and then bind to the cell surfaces of cells and be taken in by endocytosis for delivery to the cytosol and ultimately the nucleus in some cases. So you've always got your lipids, various sorts, with the nucleic acid inside. Okay, so the disease in question here is TTR, which is transthyretin amyloidosis. Okay, so this is the disease we want to treat. So a little bit about the disease. It's an autosomal dominant trait. It's caused by formation of protein aggregates basically throughout the body. This transthyretin is actually a carrier of things like vitamin A in the bloodstream, and it goes throughout the body. But strangely, this particular protein is not actually essential, so it's okay to knock it down. So the abnormal aggregates occur throughout the body, really, most commonly in the nervous system, but also heart, kidneys, and eyes. The sad part about it is that it is a progressive condition, and it leads to neuropathy of various sorts. In some people, eventually heart failure, muscle weakness, inability to walk, and early death. So this is a serious unmet medical need for this disease that we'd like to treat. Okay, so in terms of the hereditary forms of the disease, there are mutations in the protein which facilitate or make it more likely that the protein aggregates, folds incorrectly, and then forms these occlusions in the body. So starting with the folded monomer, a normal folded monomer, what's supposed to happen is that it will associate with another copy of itself to make a dimer. The dimers then associate with more copies of themselves to make a tetramer, which is the active form of this particular protein. But mistakes happen, especially when we have particular amino acid changes in the protein, which cause the original monomer to misfold, and that's the basis of the disease, that this misfolded monomer will aggregate with other copies of itself to make various other large physical structures, including fibrils and other amorphous masses of protein, which aggregate in the body, and that's what causes the problems, the accumulation of these aggregates. So this is something you want to get rid of, or at least mitigate. Okay, so this says what we do. The mutations that change the amino acid sequence cause it to misfold and form these globs of insoluble protein called amyloids and also fibrils, which will collect in the tissue and cause all kinds of problems. Okay, so the protein itself, it's produced in the liver, which is a good thing, but it circulates throughout the body. It's a small protein, and its job is to transport thyroxine and retinol, etc. So the mutant form, which is what we're trying to worry about, forms these tetramers that fold, well, the mutant form folds unstable molecules, which aggregate to form these amyloids. So what we want to do is knock down the expression of this particular protein by means of getting rid of the RNA if possible. And it isn't an essential gene because you can provide the vitamin A that's needed. So this excess of this protein is really what's causing the problem. Okay, so it's a sort of a summary of whatever. Adult onset for sure, and is eventually fatal. There are some treatments available, but not nearly as effectively as they should be. Okay, so many different mutations are known, and these vary a lot. Okay, so what we're trying to do now is cut down on this protein by reducing the RNA levels, and that sounds like a good target for an RNAi-directed approach. Okay, here's a little blurb from the manufacturer that says it's a generally unmet need and touts the lipid nanoparticle technology as an excellent way to be delivering this type of drug. One thing that's occurred fairly recently is that this particular type of amyloidosis has been recognized as a common form of non-genetic heart failure. So it's not a really weirdly rare thing. So the same treatment might end up being more generally useful as we recognize cases where the heart failure has a component of this protein aggregation in it, which is good. All right, so we're going to use a double-stranded RNA that's modified. That's but also as a second experimental thing in cells. They're going to use an DNA oligonucleotide that's complementary to the 3' region, and we're going to see how they work in cell lines to see what the effects are on the TTR mRNA levels. Okay, so here's an example in cell lines where we're looking at the percent starting material on untreated cells, where we're setting whatever that amount of RNA is as being 100% basically. So the treated cells are ones where we've delivered either the double-stranded RNA, the siTTR, or the TTR DNA oligonucleotide to those cells, and then measured the relative amount of RNA there after a certain time. So the bar graphs represent the repression or lack thereof of this degradation, really, of this RNA in response to these treatments. So we can see that the cells that have been treated with either the single-stranded DNA or the double-stranded RNA, the target RNA, which has been should have bind by its 3' end, actually is reduced. So it seems to be working pretty well in cells at the RNA level. So we can conclude that the siTTR RNA reduces the TTR mRNA in the cell line. The DNA oligonucleotide complementary to the same part of the TTR mRNA reduces the TTR mRNA in the liver cell line. A and B are both true. Well, A and B are both true. Right. This experiment involved RNA isolation and qPCR, protein isolation and qPCR, RNA isolation and Western blotting, protein isolation and Western blotting. Okay, so this was RNA isolation and qPCR, because we're looking at the amount of RNA, not protein. Okay, so we expect that if we've reduced the amount of RNA, we will also reduce the level of protein that's present. So this is again still in cell lines, and what we're doing is isolating the protein from the cell line, and then doing Western blots on the untreated versus the silencing TTR versus the double strand of the DNA oligo, ASO TTR. So in this Western blot, now they're not showing us any controls, which is really rude. In the untreated cells, we can actually see two bands when the Western is probed with antibodies to the TTR protein. They don't really explain why it dimerizes, but it's clear that it did, because we see two bands. But we can see prodigious amounts of the TTR protein in the untreated cells. By comparison, the cells that have been treated with either the silencing RNA or the DNA oligo binding to the same region, there's greatly reduced amounts of the protein in those cells. So it seems to be working at the RNA level and also at the protein level in cell lines. In this experiment, we are seeing light emitted by the primary antibody, secondary antibody, enzyme that is linked to the secondary antibody, chemiluminescent product of the enzyme. Right, when we see a Western blot, what we're ultimately seeing is the chemiluminescence that makes an image on a film or equivalent imaging system. Good. So far everything looks good. We compare the bands of our protein on our Western, and we can see that there is measurably decreased amounts of protein as hoped in this sort of situation. Okay, so here's clinicaltrials.gov. Most, if not all, clinical trials are actually registered at the government. If anybody wants to look for a disease, it's kind of a cool website. But that's where this information can be found. Okay, phase 2, in fact, phase 1 is often done on healthy people. Phase 2 focuses more on safety, but then there are assessments as to efficacy which is to say whether or not it actually works. And phase 3, of course, is with the larger number of patients, of course, monitoring safety as well, but with the eye towards whether this is actually working or not. All right, so this is results of phase 3 clinical trials with the drug that they're now calling Paterasan, and that was published in 2018. So this was delivered to 225 patients as lipid nanoparticles by intravenous infusion every 3 weeks. And the control patients got a placebo. And what they did was they measured the amount of, or concentration of, I guess, serum thyroidine over 18 months in both the treated and untreated control groups. Now, of course, what we hope is the serum transthyretin protein will be reduced, and this will prevent further formation of the amyloid deposits in the body. We also hope, but there's no reason to necessarily assume this will happen, that the existing deposits will be cleared and that the people will regain their mobility and that their quality of life will increase. So here's the results of the study. The transthyretin did drop a lot in treated patients and remained low over the course of 81 weeks, which is kind of impressive. So we're measuring the amount of serum thyroidine, which is to say we're measuring the protein, and in the untreated people, of course, the levels in the serum didn't change, the people given the placebo, but the people given the Paterasan, within a very short amount of time, the amount of circulating serum transthyretin plummeted in exactly the way one might hope and remained low for the duration of the study. The samples in the above graph were probably analyzed by Western blotting, sequencing, qPCR, fluorescence microscopy. Western blotting, of course. Okay, so we conclude that the treatment worked to reduce the transthyretin protein in serum of patients. Right? The treated people were consistently reduced, and it kept being reduced. So the hope is, not only is that the case, if they would maybe see some improvement, and that maybe the body can actually clear some of the deposits. But in any case, this is a progressive disease, so if it simply stops the disease in its tracks, meaning that it's not getting any worse, that's still a huge achievement. Okay, so did it work? People were evaluated at the beginning and the end in a lot of assessments of impairment. People that have the disease can have difficulty walking. They may have motor weakness, high blood pressure, various types of neuropathy, inability to feel and taste things, whatever, quality of life, weight loss, etc. There's a battery of tests that were applied to these patients. And so these results overall, like the line at the top with the placebo, the quality of life score, both types, got worse, because higher means worse in this particular assessment. So the untreated people during the 18 months actually suffered a lower quality of life given the progressive nature of this disease. But the people on the drug, the Paterasan, it at least didn't get any worse, and in some cases actually improved significantly, which is really good news. So that means it's really a good drug, and this is going to be very helpful to people. So FDA approval soon followed, and this was the first RNA eye drug to get FDA approval. So maybe people who have the disease can get treated earlier, not to wait until they have a serious progression. That would be something we would hope for. And it also might be useful for people with various other types of neuropathy of one sort or another, because these amyloids do occur in people that do not have the genetic defect.